as Kelly said, my lab works predominantly on plant evolutionary genetics. We everything from sort of transposable elements and genome evolution to coevolution between plants and microbes in the field. We work mostly on maize, although we dabble in a few other plants. And we spend a lot of time thinking about genetic diversity in maize. And what I will walk you through today is been sort of several years of work thinking about the origins of some of that genetic diversity and the contributions of wild relatives to modern maize. Maize sort of first captured my heart, as Kelly alluded to, when I was doing work as an ethnobotanist during my master's degree, just because of the sort of incredible phenotypic diversity in maize and the beautiful array of not just cob phenotypes, but also plant architecture and a bunch of other phenotypic diversity. But this just sort of tremendous phenotypic diversity really captivated my interest. And I soon realized that, of course, maize has not just phenotypic diversity, but tremendous genetic diversity as well. And this is maybe my favorite way of showing that graphically. This is a fluorescent in situ hybridization of some of our teosinte plants done by Jim Birchler's lab. The different colors just represent different kinds of repetitive DNA, different transposable elements, and simple satellite repeats across the genome. And each row here is a different individual of teosinte, the wild relative of maize, showing both copies of each of the 10 chromosomes of maize. And you can really see the sort of impressive diversity, not just heterozygosity within an individual, but diversity among individuals. And we actually have representatives here of multiple different populations. So there's really a tremendous amount of genetic diversity in maize as well. And it's this genetic diversity that has allowed maize to adapt to a really stunning array of environments. In fact, of all of the major crops, maize has adapted to a broader geographic area or a broader region across the globe than any other major crop, all the way from southern Finland to the equator to extreme latitudes in the southern hemisphere. And in fact, maize is grown in lots of places where you think that it shouldn't make much sense. So this is a Hopi maize field in the Mojave Desert, where although it's growing in the desert, maize is grown in rain-fed conditions without irrigation. And you can see these very sort of unusual looking maize plants with this very low density planting. And maize grows just fine up to extremely high altitudes. So this is a maize field in the Andes at above 3,000 meters above sea level. And what I'll walk you through today is sort of our work on this genetic diversity in maize and a recent sort of surprising finding about the origin of what we think is the origin of all modern corn. So because of its unique morphology, we're all probably familiar with what maize or corn looks like on the left. And because of its pretty striking morphology, the origins of maize were pretty hotly debated for a long time. Maize looked very, very different from sort of your standard canonical grass, or what we most think of as our idea type of what a grass looks like, with this giant ear and a single stalk. And this extreme morphological difference led to quite a bit of confusion and debate about the origins of maize. Maize even looks very different from its closest congeners. What we know now are teosinte, which is other wild species in the genus Zea. Maize are shown on the right, teosinte on the left. Maize differs in a number of morphological respects from teosinte, from branching architecture. So teosinte has these many separate stalks or tillers. Teosinte also has lateral branches. And the lateral branches in teosinte end in a male inflorescence, shown by this arrow on the left, and the lateral branches in maize are extremely shortened and terminate in a female inflorescence or an ear. Of course, one of the most striking differences between maize and teosinte is the ear itself. An ear of maize shown on the right of sweet corn, and on the left is a pretty typical teosinte ear, which is a single rank of 6 to 10 kernels. This is an immature ear, so this is just sort of blown up, showing each of these individual kernels. And the kernels in teosinte are inside. What you're actually seeing here is a fruit case which is a hard structure. When it matures, is a very hard structure which disarticulates, and so the ear shatters and disperses the seeds. So this is a sort of a cartoon of the fruit case, and then the actual kernel, the bit that you could eat, is inside that fruit case. So to get at the kernel in teosinte, you'd have to break open this hard fruit case. So there's really striking differences, and it's not immediately obvious the relationship between these taxa, and this led to quite a bit of confusion in the origin of modern maize. So much so that even through most of the 20th century, there was, well, more than 100 years of debate about the origins of maize. And even midway through the 20th century, prominent botanists like Edgar Anderson still questioned whether maize came from the Old World or the New World, and debated perhaps maize originated in Southeast Asia. Throughout most of the 20th century, the prevailing hypothesis was one led by Mangelsdorf and his students. That was a sort of strange, in retrospect, hypothesis that maize actually originated from a hybridization event between some perennial grass and an extinct maize, of which now there is no evidence anywhere. So some extinct maize hybridized with perennial grass, and that led to both modern maize that we know on the left and the wild relatives that are the extant wild relatives today, teosinte. And so the idea was that both of these taxa originated from this hybridization event. What we know now was the correct answer was first formulated by George Beadle, actually during his PhD work in the 1930s, but he later sort of rekindled efforts at addressing this hypothesis later in his career. So George, after his PhD, he had a mostly uneventful career on fungi and fruit flies, for which he was awarded a Nobel Prize. But after that, he came back to his true love in retirement, doing quite a bit of work on the genetics of maize and teosinte, using crosses between maize and teosinte, looking at careful look at the anatomy, looking at cytology and homology among chromosomes, and even cooking. So this is George on the left, showing that even the toughest problems can be solved by a good tortilla. He made both pop teosinte, so you can pop the kernels of teosinte in the fire, and made tortillas from teosinte. And what George argued, and we know now is the correct answer, that instead of teosinte and maize being originated from some purported hybridization event between extinct and or imagined taxa, teosinte itself, the extant wild teosinte, is the direct wild ancestor of modern maize. But teosinte, as I've alluded to, is a broad term referring to all of the taxa in the genus Zea. And so this is showing most of the taxa in Zea. There are a number of wild species. They vary in a bunch of different phenotypes and adaptation to different environments. Some are perennial and distributed across Mesoamerica. It was pretty readily apparent to most workers that the probable direct ancestor of these teosintes, the probable direct ancestors of modern maize, were one of these two annual subspecies of Zea maize. So either Zea maize subspecies parviglumis, shown on the left, and throughout the talk I'll use this red icon and red color for subspecies parviglumis, or subspecies mexicana, which is shown in blue here with this blue icon. These two subspecies diverged something like 60,000 generations ago, and so that's somewhat equivalent. It's about similar time frame in terms of units of effective population size to the divergence between humans and Neanderthals. Mexicana, shown in blue, is adapted to high elevations of the Central Plateau in Mexico, so occurring above about 1,800 meters. And parviglumis is adapted to the low, wetter tropical regions of the lowlands of Southwest Mexico. They differ in a number of phenotypes, from stem coloration and hair density to flowering time, tassel architecture, et cetera, most of which are thought to be adaptations to the respective highland and lowland environments. Mexicana is generally found in small, sort of weedy populations in disturbed habitats in and around cornfields in high elevation. So there's a Mexicana on the left with my colleague Ruben as a metric for size. Parviglumis is found in, or at least historically was found in much larger continuous populations with census sizes often in the millions of individuals. So this sort of lawn of grass you see on the right is all teosinte, and you can find just hillside after hillside covered in teosinte, historically anyway. That has changed quite a bit in the last 30 to 40 years because of anthropogenic disturbance. And so most workers realized there was one of these annual taxa that was probably the direct ancestor of maize, but we didn't really understand how maize arose from those. And work from John Dobley's lab sought to refine this understanding. So John, in the early 2000s, used hundreds of microsatellites and genotyped a number of maize and teosinte, and found, built the tree shown on the left, and found that all of modern maize groups as a single clade nested within Parviglumis, arguing pretty decisively that maize originated directly from subspecies Parviglumis and that there was a single origin of modern maize. And this paper has been very widely cited and was sort of the poster child for a simple single domestication. So maize is always held up as this nice example of a very clean maize was domesticated once in one location in Southwest Mexico from subspecies Parviglumis. And this genetic work, combined with archaeology, led us to this understanding of, at least until very recently, to this understanding of maize origin and spread. So I'm showing Parviglumis here in left, the natural distribution of Parviglumis, and Mexicana here in blue. Maize was domesticated in the Balsas region of Southwest Mexico. We know from archaeology about 9,000 generations ago, or 9,000 years ago, excuse me. Then relatively rapidly spread through the lowlands of Central America to South America, where it's found as early as 7,500 years ago in Panama, and the Peruvian lowlands 6,600 years ago, and then spread to the Andean highlands relatively quickly. Subsequently spread to the Southwest US, to the highlands of the Southwest US, and from there to northern and southeastern US, eventually to Europe, and then only very recently the sort of admixture event that we refer to as the Corn Belt dent. So most of the maize that is grown throughout the Midwest US and is as a result of this more recent hybridization event between southern dents and northern flints. And sort of this is where my lab comes in, because even in the first reading of John Dobley's paper on the origin of maize, there was a fairly evident paradox in his data. And John pointed out that the microsatellite data suggests that actually the maize, the maize that look most genetically similar to Parviglumis, if we assume that Parviglumis is the direct wild ancestor, the maize that is most genetically similar to Parviglumis actually comes from the highlands of Central America, Mexico, sort of where this arrow is pointing. Thus, that there's a geographic gap between what looks like the most ancestral, or the maize that looks most like teosinte is not found, most like Parviglumis, is not found where Parviglumis currently grows, and is not found where the archaeological data suggests was the origin of maize, or domestication. So that sort of geographical disconnect was somewhat surprising. And so we worked a number of years ago now with John to try to address this. We sampled, at the time, was a large number of SNPs from a very large sample of maize and landrace individuals, maize and teosinte individuals from across the Americas. We sampled landraces, which are these traditional open pollinated varieties. So we're not talking about the inbred or hybrid varieties currently grown in modern fields. These are traditionally cultivated varieties. And we genotyped these for 1,200 SNPs. And we just looked at initially at population structure. So this is a typical structure plot. So each column is an individual, and the colors represent what proportion of its genome is attributed to each of these different teosinte taxa or maize. And within each group here, I've ordered the individuals by their altitude shown above the graph. And you can see if we just look at these two teosinte, we see evidence that Mexicana individuals at low elevations and Parviglumis individuals at high elevations appear to hybridize or at least have mixed ancestry. And we know now that there are, in fact, hybrid populations or admixture zones between these two taxa. And then when we look at modern maize or maize landraces, we see that in the South, in the Caribbean, and also South America, although it's not plotted here, we see very clear single consistent population structure of maize separated from teosinte. But as soon as we get into the maize from western Mexico and central Mexico, we see that as we increase in altitude, as soon as we get to an altitude above which Mexicana occurs, showed by this dotted line, we start seeing evidence of admixture between maize and this additional teosinte mexicana. So maize is purported to have evolved directly from Parviglumis, but here we see evidence of admixture with the second teosinte mexicana. And we see that in the highlands of central Mexico, as much as 20 to 40% of the genome of maize landraces comes from the second teosinte. And we asked, could it be that this admixture is what's throwing off Don's data and making maize from the highlands look more like Parviglumis? And so what we did is we took a spatially explicit approach where we estimate sort of an allele frequency surface across the map and ask which point on the map, first to replicate John's results, which points on the map look most like the direct ancestor of maize. And when we do that, we get a result very similar to John's. So the black dots here show the regions on the map, the sampling locations on the map that are most genetically similar to the direct wild ancestor Parviglumis. And we see that, again, they come from this geographic area that is not where Parviglumis grows. They're in the central highlands of Mexico above 1,700 meters. Instead of comparing directly to Parviglumis, we said, what if we reconstruct what we think is the ancestral set of allele frequencies in maize? And to do so, we just used all maize individuals and effectively took the average allele frequency from maize. There's a little bit more involved in that, but essentially we're looking at the average allele frequency of maize and testing that as the sort of ancestral maize. And when we compare places across the map to this reconstructed ancestral maize frequencies, we find a different picture where regions in the highlands of central Mexico actually look much more different from the sort of reconstructed ancestral frequencies, and regions in the southwest lowlands, the black dots here along the coast, right within the geographic distribution of Parviglumis and near archaeological sites, are now the sampling locations that look most like these reconstructed ancestral frequencies. So by comparing to this reconstructed frequencies, we actually sort of recapitulate what ecology and archaeology suggests, that the most ancestral looking maize comes from southwest Mexico at reasonable altitudes within the range of Parviglumis. But it was clear from that, so that suggested to us that perhaps admixture between Mexicana and highland maize was making highland maize look more like teosinte than one would expect. But it was clear also that this admixture warranted a closer look. We know from archaeology that maize made it to the highlands of central Mexico about 6,000 years ago. But by being in the same region as Mexicana, I don't just mean sort of broadly sympatric, but these actually grow extremely close together. So shown here is a maize field on the left, and on the right, a few individuals of this teosinte Mexicana growing just meters away from the maize field. And these hybridize frequently in the field. You can go out and collect F1 hybrids all the time. And in fact, the hybrids often, especially as young plants, are indistinguishable from maize and will pop up within maize fields, as it's very difficult for farmers to weed them out. Not only do these grow sympatrically, but highland maize actually shares a number of phenotypes with Mexicana. So I showed you these differences in the stem phenotype between Mexicana and Parviglumis, the highland and lowland teosinte, where the highland teosinte has these dark red stems and these dense macrohairs, both of which are thought to be adaptations to colder environments. Highland maize and lowland maize show similar differences, where the highland maize has these phenotypes much more like highland teosinte, also shares flowering time and a number of other phenotypes with the highland teosinte, suggesting to us that there may be something more than just sort of neutral admixture and that there may be adaptive introgression going on. And we've looked at this a couple of different ways, most recently in a preprint just put online last week, that was the work of Aaron Calfee, a graduate student here at UC Davis. And Aaron, and what we did is we sequenced about 350 individuals from 14 sympatric population pairs to just really low coverage sequencing. So all of the analysis was done on genotype likelihoods. And these population pairs shown on the map really span the altitudinal range of Mexicana, shown in pink, there on the histogram on the top. And first, we just estimated for each individual what percent Mexicana ancestry in all of our individuals. And we see that both in maize, shown on the bottom, and Mexicana on the top, we get this nice correlation between Mexicana ancestry and elevation. So consistent with our earlier results, that maize, as you go up in altitude, maize gets more and more of its ancestry from the second teosinte Mexicana, so much that some individuals at the highest elevation are 30 to 40% Mexicana. We see the same pattern of Mexicana, so that you get less maize ancestry or more Mexicana ancestry in teosinte as you go up in altitude, suggesting to us that perhaps this sort of decline in ancestry, suggesting that there was perhaps selection for more teosinte ancestry at high elevation. We first asked the question about the origin of this ancestry. Was it local and sympatric? So if our green sort of circle here is our focal maize population, is most of the Mexicana ancestry coming from its sympatric next door teosinte population? Or is this sort of older global or ancient ancestry that came from multiple different Mexicana populations? And so we used an ancestry HMM to go along the genome of each individual and identify regions of the genome that come from either maize or Mexicana. And we identified Mexicana ancestry within each individual, and then asked if we just look both in maize and in teosinte, if we just look at regions that are teosinte ancestry and calculate FST, can we distinguish between these hypotheses? So that's what I'm showing here. Each row and column is a different population of either maize or teosinte. And we're only looking at FST in those regions of the genome that are homozygous for teosinte ancestry, either teosinte ancestry in a maize genome or teosinte ancestry in a teosinte genome. And first we see that the Mexicana ancestry in maize is actually more similar to other maize populations than it is to any Mexicana population, suggesting a common origin of the Mexicana ancestry in all this highland maize. We also see, if we look at sympatric populations shown by the white dot here, we see essentially no real difference in FST between maize and its sympatric teosinte population and any other teosinte population, suggesting very little evidence for recent or sympatric gene flow, and instead being consistent with most of the teosinte ancestry we see in highland maize having an older origin. In fact, when we estimate the age of this ancestry, we get an age much older than 1,000 generations ago. And when we look at correlations between recombination and ancestry, we see this nice positive correlation across all of our populations, suggesting this kind of a positive correlation where we see less gene flow from teosinte in regions of low recombination is consistent with selection against recent introgression. So both our estimated age and these FST patterns, as well as this correlation between recombination and ancestry, all suggest that there isn't a lot of very recent introgression from Mexicana, and that most of the Mexicana ancestry we see is relatively old and may have come from multiple different Mexicana populations. Still, we see lots of Mexicana ancestry, and we're interested in identifying regions of the genome that may have more Mexicana ancestry than we would expect. And Erin pioneered a really nice approach, a model-based approach, where she built a variance-covariance matrix, not of allele frequencies, but of ancestry frequencies across populations, and used a multivariate normal sort of model to predict the expected amount of ancestry we should see at any region of the genome in any given population. And so we can use that expectation to look for outliers or regions of the genome where we see more Mexicana ancestry than expected. And so that's what I'm showing on the bottom plot here, is along the genome on the x-axis, and the mean Mexicana ancestry across all populations. And anything above that blue line, that's a 5% false discovery rate based on our sort of model-based analysis of how much ancestry we should expect to see. And we can find a bunch of regions in the genome, flowering time and other candidates that have more Mexicana ancestry than we expect by chance. We can also do an analysis where not just the total abundance of Mexicana ancestry, but do we see a correlation between Mexicana ancestry and elevation that's stronger than we expect by chance. And we find a number of regions that pop out here, including this very obvious peak on chromosome 4, which is a known inversion polymorphism. And across the board, if we look at all of these candidates and ask, do we see some patterns consistent with the idea of adaptive introgression, sort of the most telling comparison is if we look at these and compare them to QTL for phenotypic differences between the highland and lowland teosinte. And so this was mapping that was done by John Dobley's lab a number of years ago, where he just mapped a bunch of these phenotypic traits that are thought to be adaptive between the two teosinte subspecies. And we find that 6 of the 9 QTL that overlap phenotypic differences between these are found to be ancestry outliers in our data, suggesting that the loci that underlie adaptive phenotypes that differentiate the two subspecies are enriched in regions that have introgressed from the highland teosinte into modern maize. And they include a number of candidate loci, such as on chromosome 9, there's a novel inversion we discovered that includes a locus that has been cloned for macrohair density, a locus on chromosome 2 that has been implicated for stem pigmentation, and this large inversion on chromosome 4, which has been implicated in a number of different traits. So overall, this suggested to us pretty strong evidence that there's that introgression or admixture between Mexicana and highland maize has led to sort of adaptive evolution in highland maize, or allowed maize to adapt to the highlands. So if highland adaptation to the highland environments was facilitated by admixture with Mexicana, we then asked, maybe has this admixture played a wider role? And we started with a collaboration with Jutta Fonseca and Tom Gilbert at Copenhagen to look at the origin of maize in the US. So archaeological data suggests that maize in the US, maize made it to the US about 4,500 years ago to the highlands of Southwest Mexico. But archaeological literature sort of debated how maize got there. And there are 2 possible routes, either a coastal route through the lowlands or a highland route directly through from the central plateau. And to look at this, what Jutta did was we used samples collected from Tularosa Cave using sequence capture. So we sampled a number of archaeological samples, 32 samples dating from 750 years ago to more than 4,000 years ago, and compared DNA from these to 2 modern landrace genomes. And we did sort of the simplest sort of tree-based analysis using Patterson's D to compare our hypothesized tree, where maize in the Southwest US originated in the highlands of central Mexico, to alternative trees where there was either an ancient origin of US maize, or US maize had a lowland origin coming up the coast. And so we can just ask which of these trees is most consistent with the data with the statistic like Patterson's D, which is just looking at patterns of derived allele counts in the different tips on the tree. And if we look at our oldest ancient sample from 3,000 years ago that had good DNA, we see that Patterson's D is right around 0, consistent with our hypothesized tree of a highland origin. But as soon as we look at our more recent samples, 2000 to modern samples, we see a very positive Patterson's D consistent with this lowland origin tree, suggesting that we had a highland origin of maize in the Southwest, followed by subsequent gene flow along the coast. And consistent with this, if we look at sort of admixture patterns between lowland maize and teosinte and these Tularosa samples, we see that consistent with with what I just showed you, highland maize in central Mexico has lots of teosinte ancestry, shown in dark brown. Our oldest archaeological sample has quite a bit of teosinte ancestry from Mexicana, consistent with a highland origin. So highland maize that had admixed with this second teosinte Mexicana, having moved up the central coast. And then as we get farther or closer towards the present, gene flow from lowland maize, this sort of light tan, we get additional gene flow from lowland maize, and the Mexicana ancestry sort of decreases through time. consistent with a highland origin followed by subsequent gene flow from coastal regions. So if highland maize in the southwest US also has some Mexicana contribution, so the second teosintes contributed to maize in the US, we then asked, what about other highland populations? So maize is found in highland populations in lots of other places. And we thought, well, what about the Andes in South America? So there are no teosinte in South America. So in the Andean populations, there's no possibility of modern or recent admixture between maize and teosinte in South America. And the archaeological data shows really clearly that maize was in Panama, in the lowlands of Panama, 7,500 years ago, well before maize ever even made it to the highlands of Central America. So maize was already in Panama before any admixture with teosinte, suggesting it at face value, at least, that this Mexicana teosinte couldn't have contributed to highland maize in the Andes. Nonetheless, highland maize in Mexico and highland maize in the Andes show a number of convergent phenotypes, from ear height and tassel branches to flowering time to stem pigmentation, and some evidence for macro hairs as well. So a bunch of convergent phenotypic adaptations to highland environments, which led us to ask, well, what is the origin of these this phenotypic convergence, and is there any evidence of a contribution from highland maize in Mexico? And so work by former postdoc Li Wang, who's now at the Shenzhen Ag Institute, and we posted this preprint late last year. We used some published data from another one of Li's studies of 36 high coverage maize and teosinte genomes sampled from low and high elevation populations in Mexico and Guatemala and South America, and did a fairly simple sort of genome scan approach where we used this PBE statistic, which is essentially a branch-specific FST, asking for FST outliers or allele frequency outliers on a single branch of the tree, using the direct ancestor Parviglumis as the third branch. So we compare Mexican highland to Mexican lowland populations looking for evidence of selection, and we do the same thing in the Andes. And we find that while most outliers are like this arrow shown here, are found only in one population. So this outlier or evidence of selection is found unique to the Andes. And most of the selection we see in both Mexico and the Andes is unique to one population. Nonetheless, about 10% of the loci that show evidence of selection seem to be the same locus showing evidence of selection in both populations, where both highland populations show evidence of selection when compared to their respective lowland populations. And we then set about asking, well, what's the origin of this convergent evolution at the level of individual loci? And we used a nice approach from Kristen Lee and Graham Coop, which takes sort of a simple tree-like structure of populations. So you build a population tree, and then at any individual selected site, you use the covariance in allele frequencies moving away from that selected site to differentiate between a number of different models of the origin of that selected site. And so the approach allows you to distinguish the probability of independent mutation, so the selected site mutated independently in each population, whether they inherited the selected allele from standing genetic variation in the common ancestor, or whether the site originated in one population and migrated from one population to the other population. So looking at these patterns of covariance, we can get at these different models of convergent evolution. And again, although most of the loci show evidence of selection in only one population, of the loci that show convergent evidence of selection in both populations, somewhat to our surprise, we found that the vast majority of these, almost 3/4 of these loci, show strong evidence of this migration model, consistent or arguing for an origin in Mexican highlands migrating to the South American highlands. And this was very surprising to us. We're not aware of any long distance recent gene flow from the Andes or from Mexico to the Andes. And in fact, previously we had done some explicit spatial modeling of the likelihood of this approach, and had suggested that migration from Mexico to the Andes was impossible. Our empirical data, of course, now suggests that our modeling was wrong, and I'd be happy to chat about what we got wrong, and it has some interesting implications for thinking about adaptive evolution that we had we were ignoring polygenic traits and thinking too much about G by E, and I'd be happy to chat about those distinctions and what we got wrong with our model. Nonetheless, the empirical data pretty clearly shows that although a minority, a meaningful proportion of the highland adaptation in the Andes arose as gene flow from the Mexican highlands. And when we look at ancestry in the Andes, we also see some evidence of this second teosinte mexicana that only occurs in the highlands of central Mexico. So although we think that Andean maize originally came to South America through a lowland route, these data suggest some contribution of subsequent gene flow from the Mexican highlands. And this led us to start thinking, well, if highland maize and the second teosinte mexicana are found in the Southwest US and in the Andes, could they be more broadly distributed? Previously, when we've looked at this, we've always used lowland maize, so maize in the lowlands of Mexico, as our comparison, assuming that there was no Mexicana ancestry in that lowland maize. And we began thinking about what's the possibility that this model is the sort of the wrong null. And we took advantage of a couple really nice collaborations to get some and large data sets to start answering this question. And so sort of the rest of this talk is unpublished work that we're working on writing up right now. In collaboration with Jianbing Yan's lab at Huaozhong Agricultural University, my former postdoc, Ning Yang, who as of a week ago is now a professor at Huaozhong, has led this study that we've termed the ZMAP study, where we've deeply sequenced about 150 teosinte genomes across all of the taxa in Zea, another about 350 modern maize landrace genomes, and using previously published data of 500 modern maize genomes. So these are inbred maize lines used in breeding. And we added to that some wonderful data on ancient maize, including published data on ancient maize, and most importantly, this sample N16, which actually was from our collaborator Miguel Vallebueno here at GMI and part of his PhD research. And this sample N16 is a genome that's a sample that's more than 6,000 years ago from Peredones, Peru. And the really nice thing about this sample was that we know it was in Peru 6,000 years ago. So it was already in Peru when maize was hybridizing with teosinte in the highlands of central Mexico. So there's no way that that Mexicana ancestry could physically be in this ancient sample. And we used these data to start asking the question about broader contribution to Mexicana to modern maize. And we first did so with sort of simple F4 statistics, similar to the Patterson's D. It's essentially asking about the consistency of the data with a simple tree. And so the tree here has maize as our red X originating from Parviglumis, and then our sister taxa Mexicana, and we used this perennial teosinte diploperennis as an outgroup. And we can calculate F4 for a number of different versions of this tree, where negative F4 values are consistent with gene flow from Mexicana to Parviglumis, and positive F4 values are consistent of admixture or gene flow from Mexicana into maize. When we look at maize from the Mexican highlands, we see a strongly positive F4, again, consistent with all the previous data. We know maize admixed with Mexicana when it got to the highlands. This is true in the Guatemala highlands, it's true in the Southwest US, it's true even in the Andes, again, consistent with what I've previously shown you, that Mexicana alleles are found in all of these different highland environments. But we also find Mexicana ancestry in the Mexican lowlands, where Mexicana doesn't exist, and in the South American lowlands, where Mexicana doesn't exist, and in these diverse lowland environments where you wouldn't think Mexicana alleles would be beneficial. We find it in ancient samples from South America, 600 to 1,000 years ago. We find it in published ancient data from Mexico as early as 5,000 years ago. And in looking across more than 500 modern inbreds, we see that every single one of those modern inbreds shows some evidence of Mexicana ancestry. And in fact, when we use an approach to try to estimate the dating of this admixture, we get huge confidence intervals around it, but our point estimate of the date of 6,000 BP is pretty consistent with the archaeological data of when maize made it to the highlands of central Mexico and hybridized with Mexicana. So the Mexicana ancestry in modern inbreds seems to be very old and to have stemmed from that initial hybridization event. In fact, of the sort of more than 900 genomes we've looked at, the only sample that shows no evidence of Mexicana ancestry is this ancient sample N16 from Peru, which is entirely consistent with simple origin from Parviglumis with no contribution of Mexicana ancestry. We can use these F4 statistics instead of just asking the consistency of the tree to build a sort of admixture graph of what we think, a simplified version of what we think maize origin may have looked like. And we get a graph that looks sort of like this, where Mexicana and Parviglumis diverged some time ago in the past. Then maize was domesticated initially from Parviglumis, a fairly deep divergence of South American maize, and that's consistent with work from Logan Kistler and a bunch of others that, and the archaeological data showing that maize made it in South America quite a long time ago and very quickly after domestication. A subsequent hybridization event between Mexicana and a replacement of existing maize, so that all modern maize originates from this hybridization event. Following that, secondary hybridization with Parviglumis, such that all modern maize is a result of these sort of origination from Parviglumis and multiple rounds of hybridization, such that all modern maize on average from this admixture graph is about 16% of its genome comes from the second highland teosinte. This graph, although cartoonish and missing some of the sort of subtleties and complexities of the real data, nonetheless fits the data pretty well. So the observed data here, these are different quartets of trees that you could make from this. The observed D statistics from those trees are shown in black, and the predicted from the graph is shown in green. So it fits the data reasonably well, suggesting that this tree is at least plausible. And alternative trees that we've tried, at least, that for different possible admixture events that could explain the data don't fit, or that could be possible at least, don't fit the data nearly as well as this tree. Nonetheless, we're worried about potential biases either in this approach or in using this ancient sample, and surprised by this result that all modern maize has 10 to 20% of its genome coming from a second teosinte. And so we took an entirely independent approach where we used an ancestry HMM. We took Parviglumis and Mexicana samples from our ZMAP data, removed any individual that showed evidence of admixture with maize or between the two teosintes, and then just modeled each individual maize genome as having parentage either from one of these two teosintes. And so we can sort of trundle along the genome. This is showing chromosome 7 in the reference genome B73, and ask what's the probability of Mexicana ancestry at any region in the genome. And you can see that across most of the chromosome, it's fairly low, but our overall average is, I think, somewhere around 10% here on this chromosome. If we sum these probabilities across our about 900 maize genomes, we get an average Mexicana ancestry of 18%, very, very close to the 16% estimated with our F4 statistics. So this independent approach that doesn't use the ancient sample gets a very similar answer for the proportion of modern maize that comes from this second teosinte, distinct from the lowland ancestor of maize. So that's so what we think then is sort of the story of modern maize origin is that it was domesticated in the Balsas, moved to South America pretty early on, moved to the highlands of central Mexico about 6,300 years ago, where it hybridized with Mexicana. This hybrid maize, this Mexicana maize admix population, then spread back out of the highlands, replacing maize everywhere on the map, such that even now today, maize in the Andes and maize in the lowlands of South America all have some Mexicana ancestry. So either replacing or admixing with maize everywhere on the map. And then, of course, subsequent divergence, entry into the Southwest US, divergence moving to Europe, et cetera. So that's cool, and we were surprised by that result. But it also led us to the question of, well, why would hybridization with this second teosinte be beneficial? What would have led that hybrid teosinte to spread across the map? And That's a good question, and I don't have answers for you yet today, but I can show you some data where we've started to look at this, where we've tried to find reasons or functional evidence to suggest that Mexicana ancestry might be beneficial. And we've done this a couple of different ways. The first is just looking across the chromosome like this from our ancestry HMM. We can identify regions like this at the tip of chromosome 7, where across 800 or 900 maize genomes, we see that 70% of maize has a Mexicana allele at this particular region in the genome. So we can identify regions that show very high amounts of Mexicana ancestry across all maize samples and ask, what are those loci and what are those doing? This particular example on the tip of chromosome 7 is a locus ZM-PRR37A, which is a known flowering time locus. This is just zooming in on that region in B73. So you can see that the peak of Mexicana ancestry is very small, as you'd expect if admixture was a long time ago, and very cleanly sort of outlines just essentially most of the coding region of the gene. We know that PRR37 is an important photoperiod gene that's in the photoperiod sensitivity pathway that helps the plant decide when to flower. We also can see that the maize samples like B73 have lower expression of this gene than parviglumis. Mexicana has lower expression of the gene than parviglumis as well, so the expression data are consistent in that sense. And lower expression of this gene would lead to earlier flowering in the pathway, which is the difference we see between Mexicana and parviglumis. So the highland allele is more earlier flowering than the lowland allele. And indeed, when our collaborators at Huaozhong knocked out this gene using CRISPR, we see that the knockout allele flowers much earlier in long day conditions than the wild type allele, consistent with a decreased expression leading to earlier flowering and a potential reason that this locus was selected or it shows high levels of ancestry because of its contribution to earlier flowering in maize. In addition to looking just sort of for outliers, we took advantage of a large amount of phenotypic data from Jianbing Yan's lab, where across these 500 inbreds, they've measured a number of phenotypic traits, kernel agronomic traits, disease traits in multiple environments. And we did a simple association mapping analysis, where instead of using the SNP genotype, we use the ancestry at each locus and ask whether Mexicana ancestry contributes to phenotype. And when we do this, skip to that. When we do this, we can build sort of standard Manhattan plots with position along the genome and association with the phenotype. But now this is not whether a SNP associates with the phenotype, but whether Mexicana ancestry associates with the phenotype. And we can find some nice outliers like this one on chromosome 6, which is a known locus DGAT1. DGAT1 is important in the last step of the synthesis of triglycerides in maize. And although we don't have direct evidence of its functional relevance in maize other than increasing oil content in kernels, we know that in Arabidopsis, overexpression of this gene contributes cold hardiness or resistance to extreme low temperatures in Arabidopsis. Again, potentially consistent with an adaptive role for this locus in adapting to higher latitudes, colder temperatures, higher elevation. Finally, we've taken sort of a broader approach in saying, well, okay, maybe we don't care about individual loci. Let's just ask how much of the additive genetic variance across a bunch of different phenotypes can be attributed to Mexicana ancestry. So we essentially model the additive genetic variance of an individual phenotype as a contribution of kinship matrices, either due to all the SNPs in the genome or a separate kinship matrix built just based on comparisons of Mexicana ancestry from our HMM across all 500 of these individuals. And so we can then attribute what proportion of the heritability or the additive genetic variance is due to SNPs or versus Mexicana introgression. When we do this across a bunch of different traits, the values aren't super high, but nonetheless, it suggests that a substantial proportion of the additive genetic variance or the heritability of a bunch of different traits is due to whether or not a maize line has a Mexicana allele at particular regions in the genome. And we can find some traits like resistance to southern corn rust or kernel number per row, which is an important yield trait, or vitamin A, the ratio of beta and alpha carotenoids, where 20 to 25% of the additive genetic variance in modern inbred samples used in breeding programs is determined by Mexicana ancestry versus Parviglumis ancestry at loci across the genome, consistent with a meaningful contribution of Mexicana ancestry to phenotype, to traits relevant to sort of modern agriculture and potentially adaptive phenotypes. Okay, so I'll end there. I've walked you through evidence that although maize was domesticated from a single teosinte, Parviglumis, that a second teosinte contributed significantly to highland adaptation in Mexico, and that highland allele, that highland maize then spread to other areas across the Americas, contributing to adaptation in the Andes and Southwest US, and that in fact all modern maize today has some ancestry from that hybridization event, that admixture event in the highlands of Mexico, and that admixture event is contributed likely functionally relevant alleles that have contributed to adaptation and phenotypic diversity in modern maize. So I've talked, of course, focused on maize because that's what my lab works on. But maize is also the sort of the poster child for a simple single domestication, as I showed you from John Dobley's work at the beginning. And what I'd argue is that even in the simple poster child for single domestication, wild relatives have played an important role in contributing functional diversity to the modern crop. And this is almost certainly true in lots of other crops, from potatoes to barley to date palms. Many other modern crops exist in sympatry in at least part of their range with wild relatives, and at least some evidence exists of introgression from those wild relatives into modern crops. And so it's likely that this sort of a story of the contribution of hybridization with wild relatives leading to local adaptation and contributing to modern functional diversity is probably a pretty common one across many different crops. Lots of folks to thank for the work I showed you today, especially the preprints and unpublished work that I talked about from Li Wang, Aaron Kelfi, and Ning Yang, funding from a bunch of different agencies, folks that contributed to some of the art that I showed, previous folks in the lab, Miguel Vallebueno and Jean-Philippe Villa-Calzada for contribution of the ancient Peruvian sample. And of course, current and past members of my lab who've all contributed to thinking and helping and writing and talking about this work. And with that, I'll end there, and I'd be happy to take any questions.